first started in the lab in July, I actually was co-mentoring another student, a senior at Penn State University, and Antonio came in. One of the things that we always did is I'll give lesson plans in the morning when I get up on the chalkboard and I write out different lesson plans. And my summer student Janessa would always be asking me questions, and I wanted to give Antonio an opportunity to participate. So I would ask Antonio a question. He was so shy back then that he would just smile with his cute little dimples of his and not say anything. And finally, over the course of the summertime, Antonio he would take ridiculous amounts of notes, and he worked so hard this entire summer. By the end of the time, these last couple of weeks, that it's just been him and I. Every single question I'm asking, and bam, 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 he's giving me answers, asking me questions back, and actually learning a lot about mouse genetics. And I can honestly say at 16 years of age, he has a better understanding of genetics than I did when I was 22. And Antonio spent a lot of time working and actually did a lot of data, produced a lot of data that will actually be publishable quality in the paper that we're going to be publishing from the lab, for which he's going to be an author. And I really hope that you enjoy the talk that he's going to give in regards to his summer project.
Hi, my name is Antonio, and I work under Alexander Joyner, and I was mentored by Dr. Ben Orvis, and the title of my talk today is Analyzing Cell Type-Specific Laws of Engrailed during Cerebellar Morphogenisis. So in the Joyner lab, we specifically study the cerebellum. The adult cerebellum is a three-dimensional structure consisting of three uniquely foliated areas along the medial lateral axis, and which includes the vermis, which is the most medial part, the paravermis, which is the lateral to the vermis, and the hemisphere, which is the most lateral part of the cerebellum. But for our talk today, I would like to focus on vermis, which is located at the midline of the cerebellum. And in this region, it's foliated along the anterior posterior part, which would be considered the front of your head. So since I'm looking sideways, my forehead is the anterior, and the back of my hair would be the posterior. And along this area, we have 10 major lobules, which are these pink and purple stuff, and they're separated by these fissures, as you can see. Two major cell types exist in the cerebellum, the granule cells and the Purkinje cells. The granule cells communicate with one another. The granules and Purkinje cells communicate with one another, both during development and in the adult mouse. The Joyner lab specifically studies the Engrailed genes, Engrailed 1 and 2, which help pattern the cerebellum. The Engrailed genes are expressed in both the granules and Purkinje cells. The Engrailed genes are homeodomain transcription factors, meaning that they help play a role when DNA is transcribed into RNA. The Engrailed genes are required for cerebellar development and patterning. For my summer project, Grant and I wanted to address the following question. Does the loss of Engrailed 1 and 2 and granule cells affect and or lead to Purkinje cell defects? We hypothesized that the Engrailed function in granule cells does not affect Purkinje cell maturation and or survival. As mentioned before, granule cells express the Engrailed gene as depicted in the red. To test our hypothesis, we utilized advanced mouse genetics to remove the Engrailed gene from the granule cells, and it's represented in white. Because the granule cells now lack the Engrailed gene, and now they're genetically distinct to the wild type, we want to figure out the difference between the wild type and the mutant. So we use PCR, or polymerase chain reaction, in which we start now with the genomic DNA, which is double-stranded. The double-stranded is then linearized at 95 degrees Celsius. So now we have two single-stranded DNA. We anneal the primers or attach them to based on the nucleic acid content, and then the primers would extend throughout the DNA at 55 through 63 degrees Celsius. Once the DNA is replicated, we have two new double-stranded DNAs that were synthesized by Taq DNA polymerase at 72 degrees. So the purpose of this enzyme is that it expands the primers that we've attached. We repeat the steps again. So now we have double of what we started. So after each cycle, we will have double the PCR product that we've had. But since there's a whole bunch of DNA floating around, we want to figure out, we're not sure which one is the mutant and which one would be the wild type. So in order to do this, we use electrogel fibrosis. Well, we basically run in the gel. And because there are 4 siblings, only one of them is the one that does not express Engrailed 1 and 2 in the granule cells, which is the female P3 mouse. To test our hypothesis now, if the Purkinje cells were affected by the loss of Engrailed 1 and 2 in the granule cells, we used immunofluorescence, in which we start off with a cell. Cells express proteins in their cell membrane or any other organelle. We have primary antibodies that will recognize these proteins. In this case would be the Engrailed. The secondary antibodies are bound to GFP, which earlier was defined already as the green fluorescent proteins. So these secondary antibodies recognize the primary antibodies and they attach to it. So now using fluorescent microscopy, we are able to see the Purkinje cells. So as you can see, the staining for Purkinje cell marker Galvan reveals little or no Purkinje cell morphological defects in the wild type versus the mutant developing cerebellum. So P3, P4, and P5 stands for postnatal, which is, so this would be postnatal day 3, which is 3 days after a mouse is fully born. And P30 would be considered the adult. But for our purpose, we're just in the developing stages. However, granule cell specific loss of the engrail leads to cerebellar size and patterning defects. The loss of the engrail results in the absence of secondary fissure and lobule 8, as well as an overall decrease in cerebellar size. So as you can see, the wild type, the cerebellum is larger than the mutant. And also here we have the secondary fissure and the 8th lobule, as well as here. And as you can see in this region, we do not see the secondary nor the 8th lobule. So we've concluded that now the granule cell specific loss of engrail results in a small cerebellum size and agenesis or non-formation of the secondary and the 8th lobule. And our preliminary evidence suggests that the loss of the engrail in granule cells does not affect Purkinje cells. Now I would like to acknowledge Dr. Alex Joyner for welcoming me to her lab, Dr. Grant Orbis for mentoring me, Janessa Smith will also help mentor me, the Joyner Lab for setting a good environment, Dr. Seth, HCS staff, and you guys. Any questions?
Could you go back to your conclusion? Thank you.